Hello, welcome to the final in our machine learning seminar series, looking at examples in biological sciences. This is, as I said, the last one. Today we're going to be looking at imaging data and some feature extraction from that, using this paper down here, looking at explainable machine learning for profiling the immunological synapse and functional characterization of therapeutic antibodies. As with previous ones, this is not a journal club, but we're using it as a basis for discussions around the approaches that they've used for their machine learning within them. So what we will cover, this is again probably looking very familiar by now. I'll again just go through a very quick introduction to machine learning, just as a reminder in case you haven't seen the previous ones. A quick introduction to the paper, looking at the data sets they used and their sort of aims and objectives, go through any pre-processing that they've done, so data tidying, any feature extraction, which will be quite a big part of today, and then looking at the sort of main model that they've used, a quick introduction to how that works, and then how well their model did once trained and what further exploration they did using their results. I will not go through an example in tidy models this time for the sake of time, but I will show you how you can do the same model type that they used in tidy models in case you want to go out and use it yourself. So, what is machine learning? A typical data analysis workflow will look something like this. So you will start off with collecting some data, some raw data that you will then look at and explore, do any filtering or cleaning as required, based on your exploration. Usually, you would then formalize your results by doing some kind of statistics, so some kind of statistical test that will give you an outcome that you can then report that will give you the answer that you are hoping to get from your particular experiment that you're doing. With machine learning, rather than doing statistics as a formalization, we instead build a model. And the model can then have two outcomes. So we can use the model for reporting just to show if we can build a model from something, then clearly there's some sort of information within our predictors that is useful. But also we can use modeling to predict new data that we haven't seen before. But again, when we're doing modeling, we still need to do these first couple of steps. We can't just skip straight to the modeling without looking at our data and understanding what is happening within it so that we don't make mistakes later on. So, steps in machine learning. As I said, firstly, we have to generate our data. We have to generate data for samples where the outcome that we want to predict is known. We then pick a modeling method and build our model, which we will then evaluate and refine, and we might do many rounds of this, testing different model methods and different parameters. We can then make predictions where the outcome is not known once we're happy with our model, and test those predictions to see how useful they are biologically, or how usable they might be going forwards. There are some key differences between model types that will help us decide which model we might want to use. So we can think about the outcome type that we have. So is what we want to predict quantitative, like height, in which case we'll want to use a regression model? Is it categorical, like whether or not someone is healthy? Then that would be a classification model. Some model types can do both of these. And again, the input type of data that we have will also influence what kind of model we use. So some require all of their variables to be numeric, so we might have to convert any categorical values to numbers. There's also some models expect certain behavior for our input data. So for example, some require data to be normally distributed and on similar scales and things like that. And another big consideration is the number of measures that we have. So if you have a really massive data set, you may want to use a model that can deal with that. If you have a lot smaller data sets, then some models don't work very well, but some will then work better in those instances. So, some example models. Many of these should look familiar now if you've been to all of these sessions. So the model we're going to look at today actually isn't really on this, but it's sort of along the same lines as decision trees and random forests, as we will see in a bit. So onto the actual paper. As I said, it's looking at using machine learning to profile the immunological synapse, and then using that to try and do some functional characterization of therapeutic antibodies. So the data set that they have, as I said, they're aiming to analyze the immunological synapse. They're looking at T and B conjugates. So they've got blood samples from healthy donors. In this case, they actually only have 9 different donors, so it's not on a large number of people. And they've done 4 independent experiments. But as we will see, they end up with a large number of images that they use. A couple of their donors were used twice across different experiments, and they isolated memory CD4 T cells from each of these donors from their PBMCs and co-cultured them with SEA, which is, I didn't write it down on this slide. It's essentially a antigen, so it will cause the T and B cells to interact and create immune synapses. So they co-cultured them with activating essentially antigens with EBV transformed lymphoblastoid B cells, or BLCL as I'll call them. And they also left some of these untreated, so they didn't add this antigen here. They then passed it through an image stream, imaging flow cytometer with 5 different lasers. So they ended up with images that contained bright field, which is just a sort of general overview without any particular staining, F-actin, MHC2, CD3, p-CD3 zeta, and live dead staining. All of these are just different things that we want to look at that will help us to identify these immune synapses and also the 2 different cell types that we have, so that we can identify what each thing is. And on average, there were around 55,000 images collected per sample at a 60 times magnification. And so they ended up with an awful large number of images, 2,899,572 images from this imaging flow cytometer, with around 100 to 200 events or synapses. Within these, they also looked at different conditions. So as I said, they had plus and minus SEA, which is Staphylococcus aureus enterotoxin A, which I can't say right now, but is a super antigen essentially. And then a few different antibody types that may activate or inhibit the immune interaction going on there. We'll come back to those later on. The main ones that were used for the training of the model and the bulk of the sort of exploration that we're looking at today is the plus or minus SEA here. They had an expert immunologist who annotated a subset of the data. So for the plus or minus SEA, they annotated 1,160 and 4,061 images respectively. And they also did a couple of the other conditions to see how well it worked when applied to those. So as I said, the labeled plus or minus SEA data is what was actually used for the training and validation of their model. So they had a total of 5,221 images that were actually used for training. with actually 70% of these falling into the training data, which was 3,654, and 30% for testing their model within that. So although they started off with a crazy number of images, they didn't actually use that many in comparison for actually training their model. So image classification. The first thing you have to do, obviously, for machine learning is you need a data set that is labeled. So as I said, they had an expert immunologist who labeled their images for them. They classified cells into 9 different classes, or images into 9 different classes, I should say, based on 2 different levels. So they looked at the number of cells that were in the image, so whether there were singlets, doublets, or multiplets. And then within those looked at the type of cells and their interaction with each other, and also the presence of TCR signaling. So you can see we've got single BLCL, we've got T cells without signaling that don't have this p-CD3 zeta, T cells with signaling that do have that. And then we've got our doublets where we have both the B and the T cells, either with or without signaling and synapses. There are also some with like no cell-cell interactions that aren't really of interest in particular for this, but is still included in the training, as the hope for this is obviously that you could automate the output from the imaging flow cytometer. And so therefore you'd want to be able to include everything, and you'd want it to be able to deal with these kinds of things. So based on this, they actually tried out quite a few different model types, of which we will not go into detail on all of them. We'll pick the main one. But they also used a couple of different feature engineering strategies as well. So they ended up with 6 different approaches that they tested, which were over here. So the different feature engineering strategies they used were either using interpretable features or using an autoencoder to generate features automatically from the images, which we will come back to what those are in a second. And then they looked at different model types. So they looked at a couple of more classical supervised learning models. So they used logistic regression, which is a very simple one, and also a boosting method called XGBoost, which is what we will come back to in much more detail shortly. They also tested some deep learning models. So they used 3 different convolutional neural networks. We talked through those a few weeks ago, or a few sessions ago, I should say. So I won't go into detail on how these work, but they did also include them. And you can see over here actually that a lot of them worked relatively similarly, actually. So maybe the massive sort of increase in complexity and reduction in the level of explainability meant that they didn't really use the convolutional neural networks going forwards. Each point in here is a different iteration in a 5-fold cross-validation that they repeated 10 times. So just different versions of the same kind of model types, but with different random selections of the data set, as we've seen previously. And they decided that the interpretable features combined with the XGBoost model, which is this top one here, resulted in the best trade-off between interpretability and classification performance. So that's great, but what are either of these things and what does any of that mean? So let's start off with the feature engineering strategies. In terms of the interpretable approach, this was using another kind of software and approach, which I'm not going to go into in any detail because it's more image analysis focused. They extracted just under 300 biologically motivated features that would help them to study this immune synapse. So they looked at morphology, so looking at things like the area of the cells, the perimeter, roundness, things like that. And they looked at both the image itself with the different channels for the different stainings and also the mask of that, which basically just looks overall at the shape of it. They also looked at intensity and texture of each of the channels, so looking at the existence of a stain or not, the particular distribution of it, whether there's any particular hotspots at any point in any of the cells. Co-localization, so trying to capture similarities and differences in intensities among the different channels, so whether there's overlap between them. And they use a few different distance measures to calculate the overlap between these masks down here in different channels as well. And finally, they looked at synaptic related values, so looked at the sort of distribution of intensities within the synapse compared to the rest of the cell. So you can see here they'd sort of essentially pull out this intersection between the T cell and the BLCL, and then look at the sort of ratio of the intensity here. So the intensity in the intersection compared to the intensity of the union, so overall in the rest of the T and B cell. And they also had a couple of more general QC measures so they could filter some things out afterwards. Obviously, for each of these, they pulled out a whole load of different measures to get nearly 300 features. That clearly I'm not going to go into detail on all of them, but this is a general overview of what they're looking at with these. Because there's quite a large number of features, they also added in a feature pre-selection pipeline to try and select the most relevant ones. The first thing, this takes a few steps. So the first thing they did was look at which two features, or if any two features, were highly correlated with each other. So if they had a Pearson correlation of greater than 0.95 or less than minus 0.95, then they kept one of those two features, which they selected randomly. They then used 6 different measures to rank the features, which are these ones here. Mutual information is just another way of describing relationships between features. So it basically looks at if you have the information for one feature, how confident would you be in predicting the value for another feature just based on that? Or how much more information about that would you have compared to not having the information from the first feature? They also used a support vector machine, which we looked at a few sessions ago. Logistic regression with different regularization types, which again we looked at in the first session. They also used a random forest, which we saw last time, and XGBoost, which we're going to go into more detail shortly. For each of these, they then selected the top K number of features, which is just a value that they picked different values for at different points to test. They picked the top X number from each method and then used essentially the features that were found to be important across all of these different methods here. Finally, they then calculated a correlation matrix between the features, and then they clustered all of the correlations and randomly selected one feature per cluster. This is because many of the features are likely measuring fairly similar things, and so there's likely to be quite a lot of essentially redundancy between them, so multicollinearity, where there's sort of a lot of them are containing a similar kind of information. So they randomly selected one feature from each of these clusters to try to reduce this multicollinearity between the different features. So that gave them an output of a certain number of features that they then inputted to the different model types. The other feature selection approach that they used was something called an autoencoder. So this is a much more automatic black box kind of approach. So rather than picking any particular biologically relevant features and pulling those out from the images, you just take an image, run it through something called an encoder, which essentially tries to just pull out information from the input, make it smaller, so condense it into something called a bottleneck or a latent space. And then you can use something called a decoder to try and then rebuild essentially the original input layer. These can be used in a whole load of different settings. So there's some sort of interesting uses where, for example, you can input a black and white image and use an encoder and then a decoder to output a colored image. In this case, it was just trying to summarize automatically the features within the input layer, essentially. So it's trying to pull out the important information within the input so that it can then have an output that is slightly smaller and slightly less noisy than the input, is this general idea. So they did this for each channel separately, and in this middle point here, mapped it to a 32-dimensional vector. So they had sort of 5 of these 32-dimensional vectors, which they then sort of reconstructed into a 256, dimensional feature vector that then would have been used as the input to the model. Obviously, we don't really understand what this is capturing. So that's where it's less interpretable than just using the actual biological approach that we saw in the previous slides. The nice thing about that is that actually the interpretable features seem to work a lot better than the autoencoder anyway. So in a way, that's nice because it means that we can then keep the interpretability when we're at least inputting it to our model. So comparing just within the logistic regression, the autoencoder was sort of the worst option, so they didn't end up using that. So back to the different model types. As I said, we've already talked through CNNs previously, so we're going to have a look at XGBoost this time. And this is the one that they actually used for the most part anyway. XGBoost is essentially a shortening of extreme gradient boosting, with boosting essentially being the combination of several weak models to try and create a stronger model overall. And gradient boosting essentially putting that in the context of adding these weaker models together sequentially to gradually minimize errors and improve our predictions. So what does this actually look like? In the case of extreme gradient boost, it's essentially creating a series of short or shallow decision trees that all link together to form something called an ensemble, where each tree within this ensemble depends on the results of the previous tree. So this is different to random forest, where we had a whole load of different trees, and you would run your data through all of them and then sort of take some summary across all of your random trees. In this case, you put your data through each tree in a sort of sequential manner, and then you get a sort of weighted sum prediction from all of your trees to produce a final prediction at the end. This is an oversimplification. There are a whole load of other complexities, or this is just not necessarily an oversimplification, but it's just a small part of XGBoost, which is the sort of model prediction part. There's a whole load of other complexities to optimize the runtime of this and decrease the amount of computational power that you need to run this model, which can be used when you're training the model on very, very large data sets. So this is one of the big reasons why you see XGBoost quite a lot, I think. And what this means is that rather than these trees being built sequentially, like this might suggest, you can build trees in parallel and do a whole load of stuff all at the same time to try and reduce the runtime that this takes. Slightly more detail on this. In contrast to decision trees and random forests, XGBoost actually starts with an initial leaf. So it will start off with a value or a prediction initially, which will be some kind of average or best guess usually. That then gets inputted to the first tree. It then calculates similarity scores, which compares how close each data point is within that particular leaf to the prediction that it has at that point. And so rather than building these trees on the actual data values, it builds them on the residuals. And this similarity score is similar to the impurity score that we saw from our decision trees. If this makes no sense, then don't worry too much. It's just a sort of interesting slight difference from what we were looking at last time with our random forests. Similarly to random forests, at each point that we split our tree, we want to have the best split that we possibly can at that point. With random forests, we looked at something called Gini gain, which looked at our impurity gains essentially from each split. This time we just calculate something called gain, which uses the similarity score of the split compared to the root of that. And then we'll choose the particular split that maximizes the gain at each point. The outputs of each of these trees is scaled by something called a learning rate, or eta, before being passed on to the next tree. And so this is a way that we can optimize our model by playing around with this parameter. So if you have a really large learning rate, then you will reduce your residuals quite quickly each time. And so you will learn your training data more quickly, with the caveat that you might then end up overfitting your data. If you have a really small learning rate, then it might be that you go through a very, very large number of trees without really getting anywhere at each round. Other ways we can reduce overfitting, which may look familiar from some other previous sessions as well. Lambda we can use, which is a regularization parameter that essentially results in more harsh pruning of our trees, so we have sort of fewer levels. We can also set gamma, which is the minimum gain required in any particular split. So if you have a split that does not have a gain that meets this value, then you would just stop the tree at that point. And finally, for XGBoost, because it's this sort of boosting method where you actually want slightly weaker models in each individual step, so you don't want full decision trees that will perfectly split all of your data, you usually set the number of sort of branches or levels that each of these trees can have, which I think was somewhere around 30 quite often. But you don't want them to be full-length trees, so you will stop them before they get to that point. As XGBoost is based on these trees, we can pull out feature importance in the same way as we could, or not in the same way, but in a similar way to how we could with random forests. So pulling out classification feature importance was something they certainly did in this paper. So once it had been trained, you can use some in-model feature importance, which is based on the gain that we saw in the previous slide. These can be biased if you have correlation between particular features. It can also depend on the number of features, the pre-selection process, if you have outliers, things like that, which they mostly removed in their pre-selection. But they also did many rounds of this so that they could take an average and get a more confident estimate of how good the different features were or how important the different features were. So they split the training data randomly into 5 different folds and used 5-fold cross-validation to train their classifier 5 times, and then they repeated this 100 times, so they ended up with sort of 500 different models from their particular classification model. In each training set, they used a random number of pre-selected features. So this is the K that we were talking about a few slides ago. So they selected between 30 or 200 features for each round of training. For each feature, they calculated a series of feature importances across all of these different models, and then they took the median for each feature to try and rank them within the sort of scope of the other features. So, based on this, they found the top 7 features for detecting cell classes based on, again, this gain from the model. These were based on essentially the intensity of CD3, which is pretty much marking our T cells, the co-localization of the different markers, so MHC2 and p-CD3 zeta, the co-localization of MHC2 and CD3, which should in theory be where the B and the T cells are meeting. The cell morphology in the bright field, and also the intensity of MHC2. So that sort of suggests that the classifier is looking at the intensity of CD3 and MHC2 to detect our T and our B LCL cells in the different images, the co-localization of the markers to detect the different doublet types, and also to look at whether or not there is actual signaling, and also the cell morphology to try and help in detecting the different cell types. So it sort of makes biological sense why it's choosing these particular features of interest as well. So now they've trained the model, they looked at doing some validation, and they validated across multiple different data sets. So the first thing they did was validate on the 30% of the original data, the plus or minus SEA data that had already been labeled. So the test data that they held out, so they could have a look at that. You can see these are the correct predictions across the diagonal here, and any values outside of that are incorrect. So on the whole, it looks like it did pretty good on this test data. There were some challenges, mostly in trying to distinguish between whether there was or wasn't signaling. Seems to be the main part where there are more incorrect values. They did a second layer of validation using the leave one out donor cross-validation. So they had 9 different donors, and at each point they took out one donor and tested how well the model worked based on the other 8 donors, and looked at how well it worked there. And you can see here there is a bit of variability across them, but on the whole, you still have pretty decent model types or model evaluation metrics between the two. They then looked at more separate data sets. So this is where they added in the antibodies. I will come back to these again in more detail in a second, but this is just looking at if you use different kind of stimulation settings and sort of different experimental setups, does it still work based on that? And again, we can see for the most part, it actually does seem to do pretty well. There are some more errors cropping in on less similar cell types than we saw with just the test data. But overall, this still looks to be doing reasonably well. Again, the signaling versus not signaling is maybe causing some issues. But overall, it looks like this model that was trained on the plus or minus SEA could generalize to new situations with previously unseen antibodies, and also could work based across different donors being included or excluded from the model. So the next thing they did, now that they've trained and validated their model, is cleaned their data, which seems like it's at the wrong point. But as I said earlier, because they're sort of building this with the hope that it could automatically classify cells as an output from the imaging flow cytometer, they only excluded these things sort of at the end when they wanted to use the model for any further analysis. So based on that, the first thing they did was take out dead cells using the live dead staining. They removed any out of focus images. They removed images that had a high entropy score. So this is an output from XGBoost that essentially says that it's uncertain about the predictions. So anything that is slightly questionable, they removed. And they also removed things that don't make sense given the staining. So things that were predicted as BLCL that had low MHC2, which should be expressed by this, and also T cells that don't have much CD3 as they should. They also removed outliers using a couple of different methods. So via something called isolation forest, which essentially is a similar thing. So it's based again on trees. This time it's binary trees, and essentially anything that gets split out from the rest of the data high up in those trees is identified as an outlier. For this, they only used the top 30 features based on the importance from the XGBoost training so that it didn't take so long to run. And the other outlier detection they used was something called dbScan, which is a density-based spatial clustering of applications with noise. Essentially, they transformed their images to 2 dimensions using UMAP, standardized it, and then run this algorithm that's essentially a clustering algorithm. So any clusters that resulted from that were particularly small. So if the number of images in that cluster were less than this proportion of the total images, they were removed again. So tidying up the data right at the end, essentially. And then they did some further exploration of their model. So the first thing they did was some sort of sanity checks, I guess. So they took their trained model and applied it to the full data set. So previously they were just using the annotated subset, but now they used all of the images from the plus or minus SEA data. And they found that there was a significantly higher frequency of synapses when SEA was added. So it increased from 0.95 to 2.58, which is what you would expect when giving an antigen. This also highlights the need for high throughput techniques in this instance because you have a relatively low frequency of these synapses. And so trying to use more standard methods would potentially cause more issues, or you would need a lot more work to get the same number of data points at least. The other thing they had a look at is how many annotated samples were necessary to achieve a good classification performance. So manual annotation of images is super time consuming. And also cannot be the most fun thing to have to do with your time. So if you need fewer images to train your data, then you want to know that and be able to implement that in the future, essentially. So they found, as you can see up here, that actually using about 45% of the annotated data still gave really good results. So this is based on something called an F1 macro score, which is another kind of evaluation metric. It essentially tries to combine precision, which looks at of the predicted positive classes, how many were true positives, and recall, which it looks at of the true positives, how many were correctly predicted. So it does that for each class individually, and then the macro part comes from essentially averaging over the different class F1 scores. Essentially, if you have an F1 macro of 1, then it means you have a pretty much perfect model. So yeah, 45% of the annotated data, which was about 1,500 images, achieved this F1 macro of 0.9 compared to using the whole annotated set of which gave an F1 macro of 0.92. So you can pretty much halve the manual annotation time by only using 45% of it, and still get really very similar classification performance. So it might be that going forward, you don't need quite so many annotated samples. Following this, they then looked at these therapeutic antibodies that I've mentioned a couple times in passing. So essentially the two antibodies we're going to just have a quick look at are an activating one, so T cell bispecific or TCB antibody that targets CD3 and CD19, but basically stimulates the immune response. This, when added, led to a significant increase of doublets and multiplets frequency. So you can see over here, there are more doublets and multiplets, actually slightly fewer singlets in a couple of instances, but sort of less dramatic. And they also saw that there was stronger enrichment of F-actin and MHC2 in the synapse. So you can see over here the control versus the antibody, and there's more F-actin here and more MHC2 here. So they said this suggests that there's enhanced formation of tight immunological synapses and therefore more efficient T-cell receptor signaling, which is in line with what we already know about this particular antibody. The other one they tested was this inhibitory antibody, teplizumab, which only binds to CD3 and so dampens the T-cell responses. This was slightly more mixed in terms of the outputs, but overall seemed to reduce the frequency of the doublets and multiplets. So sort of the opposite to what we saw with the activating one, as you might expect. Again, these look to be quite smaller changes compared to the activating one. But they also saw changes again in F-actin and this time in p-CD3 zeta signaling. So it was less focused around the synapses, as you can see up here, and in the control, and more sort of distributed throughout the cells. So as we expect from an inhibitory antibody, it looks to be inhibiting the formation of synapses with these particular markers. So a quick reminder of tidy models. So this is what we've been using throughout when going through examples. It provides a more consistent interface to prepare our data, construct our models, and evaluate our results. It's sort of the middleman between packages that have been built in R to run these models and you as a user to make it easier and nicer to switch between different model types. If we wanted to use XGBoost in tidy models, then we absolutely can do that. For this, we need the engine XGBoost. So this is the package that runs the model in the background. And we can use this kind of format here, boost_tree. In the paper, they used 100 trees, so I've set that as a parameter, and we can set our engine as XGBoost and set our mode, in this case, as classification. There are a number of different tuning parameters associated with this. You don't necessarily have to specify all of these. As I said in the paper, they specifically mentioned the number of trees, which is this trees one here. But several of these I've also mentioned today. So things like the tree depth, the learning rate, minimum node size. Again, a lot of these we've seen previously as well. So you can play around with the fitting of the model and also tune some of these parameters to optimize for your own particular data set. Okay, so I thought the last thing to do would be to sort of wrap up the whole process that they made in this paper. So I sort of focused on the main model, but they also have produced their whole pipeline. So they've created this sort of open source framework, which was actually all done in Python, for use on future single cell bright field or fluorescent imaging projects. So they've made this all available, which includes the import pre-processing of the data, the feature engineering pipelines that I mentioned, so the biologically motivated feature extraction and also the autoencoder generated features. as well as the methodology for the filtering of the features and things like that. And also includes all of the machine learning that we mentioned, so the classical models, including the XGBoost, as well as the deep learning models that we mentioned as well. Obviously, we know it can be interpretable when we're using the interpretable features as the input, and also using models that are explainable, like the XGBoost that we saw. Using this on a 24 CPU machine, apparently, you can do feature extraction and class label prediction of around 250,000 images per hour, compared to a manual annotation estimate that they provided of more like 100 images per hour. So clearly can massively speed up this sort of class prediction and feature extraction compared to these more manual approaches. The one caveat with this, or not the one, but one of the big caveats with this is that everything that they trained their model on was produced from a single machine, and so would likely not be able to be used just out of the box on other data. But this is also why they've produced this whole pipeline so people can reproduce what they've done. So if they produce images on a different machine, there we can retrain or add to the training of the model to make sure that it takes into account any differences between different machines that there might be. Things like resolution, magnification, things like that. Just slight differences between different machines that you'd expect. Cool. So that's the last session in our series. Hopefully you found these useful. So yeah, thank you very much for coming.